Hello, we're continuing with my notes, cell biology and life science in a nutshell. We are learning about currently the Golgi Apparatus, which is here shown um, in the brown. So the Golgi is known as the post office of the cell because it is in charge of processing molecules for movement or transport either to various sites, specific sites where these molecules will be used within the cell, or to the outer membrane where they will be secreted to the outside environment. So the Golgi works with the endoplasmic reticulum. It gets molecules, specifically proteins and lipids and steroids, from the ER, processes these proteins and lipids, modifying them, possibly adding sugar groups or fatty acid groups, or phosphorylating them, adding phosphate groups. And these modifications mark or tag or label molecules in order to make them specific for different locations. So the molecules will be suitable for wherever they're destined. So based on their specific markers, which they receive in the Golgi, and vesicles are used, which are like sacs that bud off. So vesicles come in to the Golgi from the ER, and vesicles bud out. So on the cis phase, or the closest region to the ER, the vesicles bring in molecules. On the trans phase, or the outer phase, these vesicles take molecules out. So we saw with the various micrographs showing the transport vesicles, the cis and trans phase, and the folds of the Golgi apparatus, cisternae is what they're called. This is what the vesicles look like budding off of the Golgi. The stacks, and this is the nucleus, so the ER, then the Golgi with their vesicles. So it is basically a shipping center moving around molecules. What is in charge of. These are showing all the folds and micrographs with the stacks of Golgi and the surrounding membranes of the ER and the vesicles, etc. So the vesicles budding off here, the stacks again shown here, Golgi. So processing occurs within these stacks. So vesicles from the ref endoplasmic reticulum on the cis phase, or receiving space, on the trans phase, the shipping site, with vesicles coming out, taking molecules to their destination. So modifications involve cleavage or addition of various molecules, fatty acids, phosphate groups, sugar groups, etc., marking molecules to go to various intracellular and extracellular locations. Again, the Golgi apparatus is structurally polarized. The cis phase near the transitional region of the rough endoplasmic reticulum, a medial segment, and then a trans phase near the cell membrane. The phases are biochemically distinct. The enzymatic content of each segment is markedly different. The cis phase membranes are generally thinner than the others. As the secretory proteins move through the Golgi, a number of chemical modifications transpire. Important among these is modification of carbohydrate groups. As described above, many secretory proteins are glycosylated in the ER. In the Golgi, specific enzymes modify the oligosaccharide chains of the glycoproteins by removing certain mannose residues and adding other sugars such as galactose and sialic acid. These enzymes are known collectively as glycosidases and glycosyltransferases. Some secretory proteins will cease to be transported if their carbohydrate groups are modified incorrectly or not permitted to form. In some cases, the carbohydrate groups are necessary for the stability or activity of the protein or for the targeting of the molecule for specific destinations. Also, within the Golgi or secretory vesicles are proteases cutting many secretory proteins at specific amino acid positions. The release of proteins or other molecules from a secretory vesicle is often simulated by nervous or hormonal signal or stimulated. For example, a nerve cell impulse triggers the fusion of secretory vesicles to the membrane at the nerve terminal, where the vesicles release neurotransmitters into the synaptic cleft or the gap between nerve endings. The action is one of exocytosis. The vesicle and the cell membrane fuse, allowing the proteins and glycoproteins in the vesicle to be released to the cell exterior. So showing the folds here in 3D. Again, the folds in 3D keep common packaged proteins. That's what Golgi does. Transport from the Golgi apparatus takes place by pathways of constitutive secretory pathway. The constitutive secretory pathway operates in all cells, leading to continual unregulated protein secretion. In the absence of specific targeting signals, proteins are carried to the plasma membrane by constitutive secretion. So mostly if they have specific markers, this is what determines whether they're going to various organelles within the cell or to be excreted via the outer membrane. This is the Golgi again, 3D, very nice. The stacks. So rough ER, that's the nucleus, synthesizes proteins, packages them in vesicles, transport vesicles from rough ER, bringing proteins. Smooth ER synthesizes lipids and performs other functions, lipids, steroids, etc. Transport vesicles from smooth ER, bringing lipids. Golgi apparatus modifies lipids and proteins, sorts them to packages into vesicles, which fuse with the